Hello friends. The focus of discussion of this presentation is phagocytosis. This is listed as the third mechanism of innate immune response. So phagocytosis is a much familiar mechanism, and this is one of the most pioneer response of the immune system, right? Or the identification of phagocytosis or identification of cells performing phagocytosis has led to the development of cell mediated immunity, right? And this was done in 1880s by Eli Mechnikov, and ultimately that leads to a Nobel Prize winning discovery, right? And this is the primary method by the body to remove any foreign substances which are entering the body, especially the blood and tissue fluids, will be eliminated by the process of phagocytosis. All right. So this is the process by which an extracellular particulate material, that means a solid substance, is ingested by a specialized cell of the immune system, right? And after that will be destroyed and eliminated. That process is called phagocytosis, and the cells which are specialized for performing such reactions are called phagocytes. And there are two categories of phagocytes. One is the professional phagocytes, and the other one is the normal cells associated or cells performing phagocytosis, which are not specified for phagocytosis, but they can perform phagocytosis. So the professional phagocytes are the neutrophils and macrophages. They are the principal phagocytic cells of the body. And you have to remember that the neutrophils are the much concentrated blood cells, right? And also macrophages are present in blood and also in tissue spaces also, right? And the other phagocytic cells like Eosinophils Monocytes Dendritic cells and B lymphocytes, all of them can perform phagocytosis according to the need, right? And one of the very basic need for phagocytosis to be performed is there should be a physical contact between the phagocyte and the cell or the substance to be phagocytosed. There are various factors, various discussions for phagocytosis that we are coming through. And before that, where this phagocytosis occurs. So this has been seen throughout the body, right? Phagocytosis has been seen throughout the body, and there are phagocytic cells present in body fluids like blood and lymph. And moreover, these phagocytic cells are present in certain organs or in certain tissues also, and these cells are called fixed cells, right? And the other one in the circulation are called circulatory phagocytes. The fixed cells or the cells in the tissues or in the organs are called fixed phagocytic cells. And such cells are given certain names also, like microglial cells. When you see certain phagocytic cells in the brain, they are called microglial cells. Then alveolar macrophages when it is in lungs. Then Kupffer cells when present in liver. Then mesangial cells when kidneys, osteoclast for bones. Then peritoneal macrophages for GI tract. So these are different terminologies used to represent macrophages, or not macrophages, not only macrophages, represent the phagocytic cells. And these are the different phases or different phases or different mechanisms of phagocytosis. Initially, the phagocyte has to be activated, and then activation leads to after or usually activation is a result of an infection or an injury. And as the injury occurs, there will be chemical secretion, and these chemicals will attract the phagocytosis, and that's called chemotaxis. And the third one is the attachment of the phagocyte to the substance to be phagocytosed. Especially we are much concentrated, much interested on a microbe which is getting phagocytosed. Then these microbes are ingested. That is the ultimate phagocytosis. They are ingested by the phagocytic cell. Then The destructive stage is the fifth one, and the sixth stage is the exocytosis, the elimination process. Then we will see one by one mechanisms. The first stage is the activation of the phagocytes, and there is one more term used. It's called extravasation. Activation means usually the phagocytes present in the blood are usually involved in the mechanism for the first entry of any pathogen, and these are usually in the resting stage. They are not activated. But when you have an infection, when you have a bacterial entry into the body, the bacterial substances or the bodily secretions, either the bacterial products like bacterial proteins, capsules, lipopolysaccharide, peptidoglycan, etc., can induce or they can activate the phagocytic cell. That is one possibility. The second possibility is that as a result of a bacterial product, the body secrete or the body activates certain proteins like complement protein or the certain cytokines and prostaglandins, and these chemicals can also activate these phagocytic cells. All right. So, this you can imagine in a simple way that when you have a tissue injury, when you have a skin rupture or a skin damage, what actually happens? There is a wound, there is a damage, and there are certain chemicals released, and also there are microbes entered. And these chemicals will interact with the exterior wall of the blood vessel. They interact with the exterior wall of the blood vessel. All right. And as a result of this communication at the exterior wall, there will be certain receptor which are expressed in the interior wall of the blood vessel. Then these receptors are complementary to certain receptors on the surface of neutrophils, and neutrophils are in the circulation. It's in a flow, and when the receptors complementary to each other are coming closer, they will bind, and ultimately this binding leads to a slowdown of the flow of neutrophils, and that leads to a process called extravasation. Right, this is nothing but it's like there is a squeezing out of the neutrophils to the out of the capillaries and to the tissue sites where you have an infection. And this particular image explains the mechanisms of extravasation. I am not going to discuss in detail about this one. It's a stage by stage mechanism, like there is a rolling activation, and there is adhesion, and finally there is transendothelial migration, right? And this epithelial layer shown is nothing but the endothelial wall of the blood vessel. That means the interior wall of the blood vessel. All right. Then chemotaxis of phagocytes means is the movement of phagocytes from the circulation towards an increasing concentration of certain attractant. Already we have seen, we have discussed that there are the attractants belongs to either a bacterial in nature, like the proteins and other materials of the bacteria, or it will be a body substance like complement or chemokines or other substance, or it could be a split product, that means produced as a result of a tissue damage like kinin or other substances like prostaglandins and all, produced by the injured host cells, right? Then as a result of this, the vasodilation occurs, or there is another term also used for this vasodilation called diapedesis. That means the chemoattractants will dilate the pores in the capillaries, that means in the blood vessels, and these phagocytic cells will migrate to the site of injury or infection, and this mechanism is called diapedesis, right? So this occurs as a result of another process called vasodilation. Vasodilation is the process by which the blood vessels pores are enlarged, or the diameter of the blood vessel pores are enlarged as a result of an increased blood flow into that site. and this increased blood flow occurs as a result of the communication of the chemicals or the chemoattractant at the exterior wall of the blood vessel. Right. So this leads to vasodilation, extravasation, and diapedesis. Clear. Then this image also explains the same thing which we have already discussed. Then when you have a high concentration of chemokines, there will be more secretion of or more migration of these molecules, these cells, into the site of injury. You can see there is pathogens entered. Then from the blood you can see the migration of the substances, and ultimately there is phagocytosis. And the next stage is when the phagocytes are migrated to the site, there are the substances to be phagocytosed. Then the next stage is the attachment. That is the much needed mechanism for phagocytosis. And the attachment of the phagocyte and the material to be phagocytosed occurs in at least in two ways. One is called unenhanced attachment, and the second one is called the enhanced attachment. Right. And in the unenhanced attachment, this is actually the natural attachment. There is no external substances involved in the linkage between the microbe and the phagocyte. And for this response, there are certain receptors or there are certain molecules present on the surface of the microorganism which is going to be phagocytosed, which is collectively we call them as pathogen associated molecular patterns, or PAMPs, PAMs. Right. So these PAMs are recognized by the phagocytic receptors or the receptors on the phagocytic cells, so that they can understand that these are not self cells, these are foreign substances, and they has to be eliminated. So they come in contact with each other, they are taken in. Right. So this is unenhanced attachment. And what are these PAMs? It's shown in the box that these are the components of the common molecules such as peptidoglycan, teichoic acid, LPS, mannans, and other glucans present usually on the microbial surface or the like cell walls and membranes, which are entirely absent in human cells, so that the immune system can distinguish this is not a self cell. Right. So this is about unenhanced attachment. The second thing, the second type of attachment is enhanced attachment. Here the microorganism is attached to the phagocytic cell, and there is an intermediary molecule which connect both the microorganism and the phagocytic cell. There is a connecting bridge or a connecting molecule between these two. Right. And these molecules usually belongs to either it will be an antibody or it will be a complement protein. So these molecules will have the capacity to bind both these cells. That means at one end they can interact with the bacteria, at the other end they can interact with the phagocytic cell. Right. For example, when you talk about antibodies, these antibodies have two segments or two ends are there. One is we call them as FAB portion. One part of the antibody is FAB part, and the other part is called the FC part. So here you can see these antibodies will have an FAB portion where the antigens are bound or the bacteria will bound. That is the FAB portion, and these antibodies will be having a Y-shaped structure, and the FC portion is supposed to interact or supposed to bind by the phagocytic cells. Okay, there is a receptor called FC receptor. So this FC receptor will be bound by the FC portion of the antibody, and the FAB portion of the antibody will be bound by the bacteria. Right. So this antibody molecule here act as a connecting link between the bacteria and the phagocytic cell. Right. So this molecule here is helping in the attachment of the bacteria towards the phagocytic cells. So this antibody molecule here, it is called opsonin. right? What it is called? It is called opsonin. So this antibody act as a connecting link between the phagocyte and the material to be phagocytosed. So not only antibodies, but certain complement proteins also, like C3b can also act as opsonins. And this process of phagocytosis, by which the rate of phagocytosis is much enhanced through this kind of opsonins, are called opsonization. All right. The next step of phagocytosis is the engulfment or ingestion of the microbe, which are attached to the surface of the phagocyte. Then this process occurs as a result of an invagination formation. Right. This is facilitated by the pseudopodia of the phagocytic cells. These are nothing but the cytoplasmic extensions, and these extensions are very flexible so that they can form a layer over the microorganism. Right. And these fusion of pseudopodia encloses the material within a membrane bound structure, which is called as phagosome. And these phagosome then enter into the cytoplasm for further processing. Right. So phagosomes are nothing but the membrane-bound endocytic vesicle formed by the invagination of the membranes of the phagocytic cells around the material during phagocytic process. Right. So after the synthesis of this phagosome, a proton pump usually pump a lot of protons into the phagosome. This usually helps in an increased acidity of this phagosome. That means there is a lower pH of around 3.5 to 4. This is very crucial for the rest of the stages of the phagosome. All right. So the next phase of phagosome is the conversion of the phagosome into phagolysosome. Right. This is formed as a result of the combining of phagosome with the lysosome. Lysosome, already we have discussed about lysosome, about the features. So lysosome is nothing but an internal organelle, a membrane bound organelle within the cytoplasm of the phagocyte, which carry a lot of granules, and this granule consists of lot of toxic substances. So these membranes of the lysosome and the phagosome will fuse each other to become a single membrane structure, and this structure is called phagolysosome. Right. And the phagolysosome will create a common environment where you have the toxic substance and a susceptible microorganism. So these toxic substances are secreted in the are available in the granules, will be degranulated. That means they are released in the phagolysosome, and they will ultimately destroy these organisms or these external substances. Right. So within the phagolysosome, you can see the destruction mechanism. And not only the chemicals like lysozyme and other enzymes, there are various other killing strategies also adopted by the phagolysosome or the phagocytic cell. So how the destruction occurs? There are two possible ways of destruction. One is called oxygen dependent mechanism. The other one is called oxygen independent mechanism. The oxygen independent mechanism or that strategy covers a lot of lytic enzymes like lysozyme, glycosylase, phospholipases, nucleases, lactoferrin, and other various proteins involved in the destruction. Whereas the oxygen dependent system is usually it's depending on a metabolic process and also production of toxic oxygen derivatives. Right. So this is the this table shows you a comprised data for this oxygen dependent and oxygen independent destruction. So in case of oxygen independent process, the lysozyme is a hydrolytic enzyme which will destroy the destroy the microbial cells like peptidoglycan, and that leads to destruction. Then defense in it's a collection of or a common name used for a group of antimicrobial cytotoxic peptides. And these defensins are rich in cysteine, having some around 30 to 35 amino acids, and they can form certain channels in the membranes of the microorganism, and this helps in the destruction of this microorganism. So defense is a very important antimicrobial substance produced by most of the human body. The another one is tumor necrosis factor, TNF alpha, usually produced by macrophages. This is also a cytokine having activity against, especially against tumor cells. Then cathepsin G, lactoferrin, elastase, catholicidins, collagenase, and other various digestive enzymes all are playing vital role for the destruction of the phagocytosed cells substances. And the oxygen dependent killing, usually in which there are oxygen derivative formation, right? This occurs as a result of a respiratory burst. There is a respiratory burst which occur in the phagocytic cell during phagocytosis. This is because when the cell is going to phagocytose something, there is an increased need for energy. So the cell usually switches into a higher rate of metabolism, and then there will be a higher intake of oxygen, uptake of oxygen, and this leads to activation of more oxidase enzyme, right? And that ultimately leads to production of or release of byproducts like superoxide anions, hydroxyl radicals, hydrogen peroxides, and all those substances are reducing agents, and they can cause destruction of cellular substances, right? And moreover, myeloperoxidase can also lead to the production of hypochlorous acid. That's also toxic to the cells. You can see the reactions. There is a superoxide anion formation or hydrogen peroxide formation. All those are enzymatically mediated reactions. Then this is formation of hypochlorous acid from hydrogen peroxide as a result of myeloperoxidase activity. Then in case of macrophages, there is another enzyme called nitric oxide synthetase, which is also activated as a result of a bacterial infection. Then that leads to synthesis of nitric oxide, and this nitric oxide in turn activate or in turn combine with various substances like superoxide anion, and ultimately leads to destruction of the microorganisms. And collectively all these compounds are powerful oxidizing agents, and which can oxidize most of the chemical groups found in the proteins, enzymes, carbohydrates, DNA, lipids, and all various of these biomolecules present in the external substances like the bacteria. So they will be destroyed ultimately, and collectively all these substances, all these reactive molecules are called reactive oxygen species. So this image is a comprised image of phagocytosis. There is attachment, engulfment, the phagosome and phagolysosome formation, and the destruction. Then the final stage of phagocytosis is exocytosis. The destroyed or the lysed materials are released out of the phagocyte. That means it's a controlled mechanism, and all these process occurs inside the phagocyte, and after destruction, the destroyed substance, destroyed peptides and small molecules are released out of the phagocytic cell. That process is called exocytosis. And during this process, one of the very peculiar reaction occurs, which is called antigen presentation. And here it's a parallel reaction, that during the process of presentation, during the process of synthesis, during the process of destruction of So during the process of destruction, there is a parallel synthesis of certain proteins called MHC proteins. So these MHC proteins will combine with certain digestive peptides, and these digestive peptides are presented on these MHC molecule, and they are ultimately presented to T cells, especially T helper cells, and that leads to the activation of T helper cell. In that way, these innate mechanisms are connected to the adaptive mechanism. So this is about phagocytosis. By that we conclude the phagocytic mechanisms. So we have seen different phases like initial activation of phagocytic cell, then chemotactic attraction, then attachment, in which unenhanced attachment and enhanced attachment. Then there is destruction mediated by phagosome and phagolysosome, and ultimately the destruction of oxygen dependent killing and oxygen independent killing, and the final stage of secretion called exocytosis. Right. So that's about phagocytosis. Thank you for listening.